And so two guests, Fabrizio and Elisa. Elisa Mosconi is representing AIRC today, and AIRC is one of the best parts of our country, I would say. Is a charity, is the Italian Association for Cancer Research, as the Italians must know. And actually, the research on cancer in Italy is mostly supported by AIRC. And as we are going to see, and AIRC relies on small donations, very important, but also on a lot of volunteers, as we are going to see. Specifically, Elisa has a degree in philosophy, a laureata in filosofia, and she has always been keen on what we call the terzo settore, third sector, that is activities with charities or other social non-profit associations. And gradually that has led her to acquire a lot of experience in this field. And since 13 years, Elisa is managing the communication at AIRC, quindi è communication manager di AIRC, la cui sede è a Milano. AIRC is based in Milano. And she will at the beginning introduce AIRC and a little bit of this initiative. Next, dopo Elisa Mosconi, we are going to have Fabrizio. Fabrizio is actually, in many ways, a friend of Pavia. Why? Because his supervisor has been the famous, very famous, it's almost emotional for me, Professor Faraschi. Professor Faraschi è una persona eccezionale. Chi l'ha conosciuto lo ricorda benissimo. If you go to the room in the CNR, where with the MBG students we were supposed to have lectures, you will see a painting or depicting Faraschi. And Faraschi had a pupil named Mauro Giacca, who is also an amazing scientist. I think now he's in London, right, Fabrizio?
He's in London, yeah.
So unfortunately, Mauro moved to London from Trieste. And so Fabrizio did his PhD with Mauro Giacca, working on HIV. Quindi Fabrizio nasce come virologo molecolare. Next, Fabrizio, after his PhD, moved to Cambridge to work with, his name is Stuart, Fabrizio.
Steve Jackson.
Steve Jackson, sorry, I thought Steve Jackson, who is also a very, very well-known biologist. After Cambridge, Fabrizio moved back, and essentially now he has a double appointment, un doppio appointment, one at the CNR, so here in our campus. So some of you, especially for the MBG, may perhaps think to ask, especially Sofia, who is a co-worker of Fabrizio, about doing the laurea in Fabrizio's lab. And Fabrizio is also has a lab at IFO, that is a kind of public-private institute, top-notch guys, scientists working on cancer. And thanks to both of you. My only point that I'd like to add, also if you look at the pedigree of Fabrizio, vedete come anche nella scienza good scientists normally are, educated in excellent labs, right? Quindi when you choose your life, always think that good people make the difference. And the other message from Elisa's pedigree is that you see, you can also do science in a certain way, not only by staying in the lab, but also doing things that support science directly. And Elisa is a wonderful example. You see a person with a philosophy degree who knows a lot about cancer, and also she knows a lot about science communication. Quindi Elisa è also interessante per gli studenti per capire che ci sono vari percorsi.
Grazie Andrea.
Quindi passo la parola, I give the microphone to Elisa and then next directly to Fabrizio. By the way, last thing, so ask questions. Potete domandare domande in italiano, don't be shy. Try in English, don't be shy. Ask questions anytime, as also we do during my lectures to Fabrizio. Grazie. Ciao Elisa, it's yours.
Grazie a te, grazie ai ragazzi per essere qui oggi, grazie all'ospitalità, grazie al nostro Fabrizio per la splendida lezione che ci sta per regalare. Come diceva Andrea, appunto, io mi occupo di comunicazione per AIRC ormai da 13 anni a gennaio. AIRC è una realtà italiana con una lunghissima storia, credo che la maggior parte di voi la conosca, ci tenevo giusto a introdurre e a darvi qualche informazione puntuale. Allora, AIRC è nata nel 1965, allora era un'associazione, oggi è una fondazione, e da allora l'unico obiettivo è stato contribuire a rendere il cancro sempre più curabile. Perseguiamo questa missione attraverso due strade principali, in primis finanziando la miglior ricerca sul cancro in Italia. Considerate che ogni anno sono circa 5.000 i ricercatori che lavorano sul nostro territorio attraverso fondi della fondazione, tra cui ci sono sia Fabrizio D'Adda di Fagagna che il professor Mattevi, quindi insomma siamo in ottima compagnia, come dire. E parallelamente la missione viene perseguita anche facendo della buona comunicazione scientifica. Giusto per darvi un numero, vi posso dire che nel 2019 abbiamo destinato alla ricerca oncologica oltre 117 milioni di euro. Considerate che questi fondi sono fondi tutti privati, derivano da donazioni di sostenitori che appunto sostengono la ricerca a vario titolo. Vi dicevo, la comunicazione fa parte della missione, il progetto di oggi ne è un esempio. Noi cerchiamo di parlare al largo pubblico attraverso collaborazioni frequenti con i media generalisti, attraverso il nostro house organ che forse avete visto nelle case dei vostri nonni di solito, la pubblicazione dedicata ai nostri più fedeli sostenitori con diverse pubblicazioni, diverse copie, appunto, edizioni nel corso dell'anno. Abbiamo un sito molto attivo, degli account social altrettanto, anzi accolgo la occasione e vi invito a seguirci soprattutto su Instagram perché potreste trovare delle cose interessanti. Attraverso questi mezzi cerchiamo di portare appunto al largo pubblico i risultati della ricerca in laboratorio, ma di promuovere anche corretti stili di vita, prevenzione del cancro e di promuovere anche un approccio come dire, scientifico all'informazione. Soprattutto in questo periodo purtroppo la comunicazione scientifica ha preso una deriva non facile, sappiamo tutti quale collegamento ci sia tra il Covid e una corrente infodemia. Diciamo che è nelle nostre corde riuscire a mettere a disposizione l'esperienza di AIRC in termini di debunking e di corretta informazione e individuazione delle fonti. Questo lo facciamo anche all'università attraverso diverse lezioni dedicate proprio allo smantellamento delle fake news. Vi dicevo, questo progetto è un progetto dedicato appunto agli studenti universitari, quindi una cosa un pochino più mirata rispetto a quello che facciamo con i video generalisti. È nato con l'Università di Padova 5-6 anni fa, è stato un grossissimo esperimento, ma ha funzionato perché oggi riusciamo a collaborare con diversi atenei sul territorio italiano. L'obiettivo è raggiungere chiaramente gli studenti, è il motivo per cui siamo nelle aule, anche virtualmente come oggi, e contribuire appunto alla loro formazione attraverso case history AIRC oppure condividendo i risultati della ricerca in laboratorio, come nel caso appunto di quelli di Fabrizio D'Adda di Fagagna, direttamente con gli studenti. Quindi da una parte c'è la divulgazione, corretta informazione, dall'altra speriamo che attraverso questo progetto gli studenti possano aver voglia di entrare a far parte della causa. Questo di solito capita attraverso dei momenti più leggeri, organizziamo anche degli eventi con l'università. Andrea lo sa, quest'anno avremmo voluto partecipare alla caccia al tesoro che c'è tra collegi a Pavia, purtroppo per ovvi motivi non è stato possibile, speriamo di rifarci con il 2021. Andiamo avanti, la forza di AIRC sono chiaramente le persone, al di là dei 4.500.000 sostenitori che appunto sostengono la ricerca a vario titolo con donazioni ricorrenti, SMS, i bollettini postali, per raccogliere fondi noi facciamo tantissime iniziative di raccolta fondi appunto, le più famose sono quelle storiche come le arance della salute e la zalia della ricerca. I nostri volontari vanno nelle piazze italiane la mattina presto, una volta all'anno, e distribuiscono questi prodotti solidari. Diciamo che rispetto ad altre realtà associative siamo fortunati perché possiamo contare su 20.000 volontari, ma siamo arrivati a un punto per cui non bastano più. Le forze ci sono e ci sono ancora, ma abbiamo bisogno di nuove idee ed è il motivo per cui oggi vi chiedo se avete voglia di diventare volontari di AIRC. Diciamo che il volontariato di AIRC non è particolarmente oneroso, poi so che voi avete tanto da studiare, 1000 interessi, quindi insomma capisco. però si tratterebbe di un giorno all'anno, non di più, ma anche mezza giornata, se avete voglia appunto di seguire le iniziative di Piazza Canonica. Qui vedete una ragazza che ha più o meno la vostra età davanti a un banchetto di azzalè. Ci sono però anche delle altre possibilità, abbiamo una serie di piccole iniziative sul territorio organizzate dai nostri comitati regionali, quello lombardo a sede a Milano, poi chiaramente vi lascio tutti i contatti, oppure ci sono i social network, per cui diventando nostri supporter, condividendo i contenuti che trovate sui nostri account, oppure mettendo a disposizione un vostro talento, una vostra passione, noi riusciamo a raccogliere i fondi. Vi faccio un esempio: attraverso questa piattaforma di crowdfunding che si chiama Rete del Dono, tanti ragazzi come voi corrono partecipando a un maratone che periodicamente AIRC propone, oppure a virtual run come in questo periodo per ovvi motivi. Attraverso la loro corsa incitano i sostenitori a sostenere loro, a sostenere la ricerca, e questo diventa un modo per raccogliere donazioni. C'è poi tutta una meccanica, magari se avete voglia di maggiori informazioni io resto sicuramente a disposizione a fine della lezione. Comunque vi lascerò i miei contatti, chiederò la cortesia al professor Mattevi di condividerli con voi, e quindi se volete, insomma, noi siamo a disposizione. Questi sono i contatti appunto del nostro Comitato Lombardia, che è la sede regionale. Ultima cosa, se avete voglia di darci un feedback non solo sulla lezione di oggi, ma anche in generale sulle nostre attività legate al target, se avete voglia di dare un occhio appunto ai nostri account social, se siete già informati sulle attività di AIRC, per noi avere un vostro riscontro è molto utile, soprattutto per capire se stiamo lavorando bene, se stiamo andando nella giusta direzione. C'è un questionario di una decina di domande, una cosa molto veloce, se appunto avete voglia di compilare noi ne saremo felici. Vi saluto e vi ringrazio e lascio la parola a Fabrizio.
Thank you, Lisa. I'll try immediately to switch on the screen sharing. Let's see if that works. Do you see my slide? Yeah?
Yes.
Okay.
Sì, ti vediamo.
So thank you, Andrea, and thank you, Elisa, for having me. Again, it's always a pleasure to work with AIRC, and especially so if this is set up in as usual an excellent and efficient way by Andrea. So a very brief introduction to myself, as it has already been mentioned. I'm working both in Milan, in IFOM. IFOM, again, it's an institute which is in fact mostly, if not entirely, supported by AIRC. And so AIRC gives support to researchers throughout Italy, some also abroad, so Italians working abroad with fellowships. But again, it is also generously sponsoring activities in this research center in Milan. So I'm working there, but again, I have strong links with the Pavia as well. So I work at the CNR, at the IGM, La Nave, as some of you may know it. And again, this is also thanks to Arturo Falaschi, that has already been mentioned before. He really set up La Nave, then set up also the institute in Trieste, where I did the first degree, the biology degree, and then my PhD as well. So certainly I'm indebted to Arturo Falaschi and to AIRC, because again, after my period in Cambridge, if I came back to Italy, is also again thanks to AIRC that funded my lab in the beginning and keeps funding it. So again, if everything that I will be describing you today surely would not have been possible without the constant support by AIRC. So thanks to AIRC, thanks to Andrea, and thanks to you, but I have a request, so do ask me questions. I'll try to make this as interactive as possible. Surely it can be improved, and it can be improved by your questions. So anytime something is not clear or you want to go deeper, you ask me, I'll stop, and then we'll go deeper. I'll try to solve some of your doubts. So I'll be speaking, as you can see in the title, that surprisingly matches what you were given. And so I've been studying how cells respond to DNA damage for quite some years. And DNA damage takes place both in cancer and aging. So I'll be discussing to you today the events that lead to DNA damage generation, the consequences, and in the second part I'll also try to describe to you an approach that we recently developed in our lab that we think is maybe has a potential to not only to become an interesting tool to study how cells respond to DNA damage, but potentially it has also a therapeutic value. And so that will be the last part of my presentation today. So first thing first, what is DNA damage, what is aging, and we can probably start with the simplest, at least conceptually, experiment that has been carried out many years ago, 60 years ago. Somebody took some cells, put them in culture, they grow, and eventually they stop dividing. And it's not that you did anything wrong, you forgot to add serum, or you forgot that your cells out of the incubator. You keep your cells in the most optimal conditions to grow them, but they are being unable to further proliferate. So we say that these cells have become senescent, or they have undergone cellular senescence, which is a sort of cellular aging, or we say that cells have reached the Hayflick limit. Because Hayflick is the person that made this observation first, again, more than 60 years ago. And this is Leonard Hayflick. And I met him at a meeting in Berlin a few years back, and he's still very bitter how hard it was for him to publish his observation. So he was a guy that was saying, look, guys, I'm unable to grow myself indefinitely, while everybody else was thinking to be able to grow their cells indefinitely. So if everybody was telling him, you Lenny, you are unable to grow cells. You have a problem. It's not a result. It's a lack of a result. We grow cells forever. Your cells stop dividing. Obviously, you have a problem in being able to grow your cells. In fact, and this is just an anecdote that I want to share with you, the other people that were wrong and he was right, and the people that were saying their cell that grow forever is because they were having a very obvious artifact in their hands. So at the time, every time that you split your cells, you replenish the medium, you have, of course, to add serum, and serum is what feeds the cells for and allows them to proliferate. But at the time, you would not buy serum, you were making it yourself. And how were you making it? You were taking the blood of chicken, typically. You were filtering with a piece of cloth, and whatever was passing through the fabric, that was your serum. Problem was that serum was contaminated with new cells. So every time that people were splitting cells and adding serum, they were adding new cells. And that's why they were seeing their cells proliferating forever, just because they were adding new cells. So he was right, they were wrong, but he really had really hard times in publishing his observation, which was conceptually a very important observation, because it demonstrated that cells are mortal, meaning normal human cells are mortal. They have a limited ability to proliferate. So simple message for you guys, cynically speaking, try always to publish positive results, because negative results are very hard to sell. Trying to persuade people that something doesn't happen, it's always harder than showing a positive result. But of course, in this case, it was the right thing to show that cells cannot proliferate. So why cells eventually stop dividing? Few characteristics of these cells that are unable to proliferate. Number one, they are not dead. So cells that stop proliferating is not that they are unable to function, they are unable to proliferate, but remain metabolically active. You can quantify the fraction that proliferate or not proliferate by feeding them with BRDO, which is a DNTP analog that allows you to quantify the cells that are undergoing DNA replication or not. They have also a peculiar morphology. You may say they are fat because they don't exercise, because in fact they get larger and larger because their cytoplasm does not divide. So they keep growing, but since they don't divide, they get larger and larger and larger. And of course, there are a number of conditions that are associated with the lack of proliferation, differentiation, quiescence. So you need a marker that specifically allows you to distinguish between senescent cells from differentiated cells or quiescent cells. And there are a number of them. Nothing really works 100% of the cases. One of them is this weird enzyme. It is an enzyme that is up in senescent cells. It's a lysosomal enzyme, has no known function, but for a number of historical reasons, everybody is using this assay to show that their cells are indeed senescent. One concept that I want to introduce to you from the beginning: senescence is irreversible, meaning once a cell has pulled the brake, is unable to proliferate, cannot start proliferating again without any exogenous experimental intervention. Differently, the quiescence condition is irreversible. Quiescent cells are cells that, for instance, normal cells, they become contact inhibited. There is no room in your petri dish for cells to grow, so they stop dividing, but as soon as you split them again, there is ample space for them to grow, and they will start growing. Okay, so quiescence reversible, senescence is irreversible. So why they stop dividing? They stop dividing because, as you probably know, your DNA in your cells is broken into pieces. These pieces are known as chromosomes, and the problem with chromosomes is that they are linear, meaning they have a beginning and an end. And this is a problem because evolution so far could not come up to a solution to fully replicate linear DNA molecules. So there is still room for improvement, no? So, meaning that if this is a chromosome end, a linear DNA end, upon DNA replication, you have one strand which is fully replicated, so-called leading strand. The other strand is the lagging strand, replicated discontinuously. So you have an RNA primer, and then DNA synthesis, an RNA primer, and then DNA synthesis. You can remove this RNA, and then you can fill this gap. You can remove this RNA, you fill this gap. If you remove this RNA, you end up with only a partially replicated chromosome end. So every time that cell divide, every time that the DNA is replicated, is not replicated in full, and so the very tips of the chromosome, the telomeres, are not fully replicated. So that means that people, once they realize that, they put together the two observations, and they draw a correlation between telomere length and the inability of cells to proliferate. And so they started thinking that when telomeres become too short, this is not compatible with proliferation. That was just an association, but more and more people started to think this was an intriguing observation. That I observed that, in fact, the only cells in our body that by definition must proliferate forever, and those are the germline cells that allowed for the progeny to exist. Well, they expressed an enzyme, which is a very special enzyme. Is in fact, it's a reverse transcriptase that elongates the telomere. So it's not a standard DNA polymerase, because the standard DNA polymerase would not be able to do that, but they express this enzyme that is able to extend the very tips of the chromosome. So cells that that proliferate forever, they have it. Fibroblasts, the one that undergo senescence, do not have it, and that's why they stop dividing. Well, actually, that's why. That was, again, an association. So maybe I should stop here and ask you a question. So you are brilliant, Fabio, as a student. And so what would you do now to prove that telomeres are really the cause of this arrest, having observed that there is an additional correlation with the telomerase expression? So if you had ample means and excellent genetic manipulation skills and ample money, supported by AIRC, for instance, what would you do in order to prove that really telomeric shortening is the cause of this arrest? How would you reverse it? For instance, in the fibroblasts.
Express the enzyme.
Exactly, and this is what people did. And they got a science paper just by putting telomerase in these cells, and they observed that by just placing the telomerase gene in these cells, now these cells have become immortal, or people now say they have become telomerized, because historically immortalized cells were semi-transformed, semi-cancerous. In fact, this is not the case, but this really proved the point that telomere-initiated senescence, the replicative senescence that I showed you before, is really triggered by telomeric shortening, because just by fixing the telomere problem, cells become, in fact, immortal. So we were very happy. We thought we understood everything. And then somebody did a very simple experiment. They took human fibroblasts, human diploid fibroblasts, a piece of your skin, put in culture, or mouse embryo fibroblasts. So these are mouse cells and these are human cells, fibroblasts in both cases. You start looking at the telomeres. So the telomeres, I should have mentioned this before, they are made of repetitive sequences, TTA, GGG, TTA, GGG, TTA, GGG for kilobases and kilobases, kilobases, okay? Human telomeres are 10 kb long, mouse telomeres are much longer, 50 kb, okay? You put them in culture, and you have also to consider that these guys, I showed you before, they do not express telomerase, but surprisingly, mouse cells, most of them express telomerase. So they start with long telomeres, plus they express telomerase. You put them in culture, and these guys senesce in a couple of months. So PD stands for population doubling. So the number of times that a certain number of cells becomes double. The doubling. So you put 10 cells, the day after are 20 cells, and then become 40, then 80, blah, blah, blah. Okay. So they basically, they will keep proliferating for 1 or 2 months at least, while mouse cells, surprisingly, they age, they senesce much sooner, despite starting with longer telomeres and expressing telomerase. So when this observation was out, that was felt obviously as paradoxical. That means that people started thinking maybe we didn't understand anything, and telomeres are not the cause of cell senescence. So does that mean that everything that I told you so far is wrong? No, there is just an additional level of complexity. There was an additional mechanism that was impeding those mouse cells to proliferate. And what it was observed that mouse cells are very sensitive to the atmospheric oxygen they are grown in, meaning under our skin, we have typically 3% oxygen, but we are breathing 20% oxygen. That means to say that some cells, like mouse cells, are unable to cope with this excess of oxygen, which is an oxidative stress. And therefore, if you instead put them, so these mouse embryonic fibroblasts that they were senescing very quickly, if you grow them in an incubator with normal standard oxygen tension, you move them in an incubator that is in which there is much less oxygen, and now they start to become immortal, just as you would have expected by the fact they express telomerase, plus they start with long telomeres. So once again, another quick lesson for you. When you see a negative result that seems to upset completely what you have done before, it doesn't mean that everything was wrong. There is just an additional layer of complexity that needs to be worked out, okay? So an unexpected result, it's not necessarily negative, it's an opportunity to learn additional mechanisms. So why eventually telomeres, when they are too short, cause a cell to arrest? This is where I made a small contribution. So what we, that was my last publication when I was in Cambridge, was the discovery when telomeres, as I mentioned before, they become shorter and shorter and shorter, and each DNA replication round, eventually they become too short. And now the cells perceive them not anymore as telomeres, but as broken DNA. And when DNA breaks, it immediately triggers a so-called DNA damage response, so the response to broken DNA. And when this DNA damage response, which is a sort of alarm response in a cell, the first thing it does, it pulls the brakes to cell cycle proliferation. So it enforces what is known as a cell cycle checkpoint. So it stops the proliferation, and this is in fact the basis of cellular senescence. So we, and then others, and now this is generally accepted, propose that the replicative senescence, the one that I've showed you before, is caused by telomeric shortening, because critically short telomeres are recognized as broken DNA. DNA is extremely precious within the cell. Cells must respond by arresting proliferation. And just when you have a flat tire on the motorway, you don't keep going. You stop, and then you try to fix your broken tire. This is the same case. So you cells stop proliferating, attempt to fix it. I'll show you later they cannot fix it. And so they stay with this persistent DNA damage, triggering this alarm that keeps ringing in the cell. So with this, the DNA damage response, this is probably, seems crowded, it seems complicated. In fact, probably this is an oversimplified version of what we know. Basically, this is it's a signal transduction cascade, in which the signal is broken DNA. So when DNA breaks, you need some some proteins, some factors to sense DNA damage, to recognize there is broken DNA in the cell. So this is the job of some sensors, DNA damage sensor. This complex here, MR11 or 50MDS1 is one of them. I will not discuss into detail. So you need some sensing protein, and then once DNA damage has been sensed, you need to signal the presence of DNA damage. So the best way to signal in a cell is to phosphorylate protein substrate. So indeed, what this sensor does is to recruit a protein kinase like ATM to the sites of DNA damage, and one of the first substrate that ATM will phosphorylate is the histone variant H2AX. When H2AX is phosphorylated, we call it gamma H2AX, and this H2AX will recruit more of the sensor, that will recruit more ATM, that will phosphorylate more H2AX. So it's a positive feedback loop that leads to the formation of hundreds of kilobases of phosphorylated H2AX. So this is a massive chromatin modification event. It's so massive that you can see it under the microscope very easily. So these are nuclei of human cells that we have irradiated with X-rays. So we have broken DNA with X-rays, and you see these dots, we call them foci, and each one is an individual DNA lesion, individual DNA breaks that has been generated by X-ray, and the chromatin surrounding it has been modified, and this is recognized by antibodies against gamma H2AX. I apologize if you here see a square, but it's a gamma. So you get chromatin modification. This recruits a bunch of proteins like 53BP1, MDC1, and you see that they co-localize here. And so there is a gathering of proteins that somehow favors the signaling of presence of DNA damage and the repair of such DNA damage. Of course, these events are important locally, but of course the entire nucleus, the entire cell need to be informed about the presence of DNA damage. So there are diffusible kinases that do not just stay at the sites of DNA damage, but they diffuse throughout the nucleus, informing all relevant partners that DNA damage has been generated and something must happen. And eventually somebody must take decisions. So these are the so-called effector proteins that take decisions. And the decisions may be, I'm gonna kill myself, that's called apoptosis, or I'm gonna arrest my proliferation, either transiently or permanently. The permanent arrest is what we call, again, cellular senescence. So this seems fairly clear, linear, but then in fact, when I was putting together this slide, I realized there was something that I was missing, something that I could not understand. Why is it that the same lesion here eventually triggers two opposite outcomes? Here cells will never proliferate again. Here cells arrest, but then they proliferate again. So the same lesion eventually leads to an arrest or actual proliferation. Why is that? So when I was asking this question to my colleagues, people, they were telling me, Fabrizio, it's obvious. Because here, you get DNA damage, cells arrest, then DNA damage is being repaired, the checkpoint is switched off, and cells start to proliferate again. So I was very happy with this explanation, but the inference of this reasoning is that cell senescence instead is associated with some kind of irreparable DNA damage that cannot be repaired, and therefore the alarm keeps ringing in the cell. You have a problem, you cannot proliferate, but again, this implies the presence of irreparable DNA damage. And when I was going to DNA repair meetings where people study DNA repair, certainly the concept of DNA damage that cannot be repaired was not very popular there. So we really had to check whether was it really true that sometimes DNA breaks cannot be repaired. So we did a very simple experiment. We take some human fibroblasts, we expose them to X-rays. Again, this is really carpet bombing, and you can see how many of these gamma H2X or another marker foci we can see. And then we monitor the resolution of these foci, of these dots, in time as a proxy, as a readout of the ability of these cells to repair DNA damage. And we observed that indeed 3 days later, most lesions have been repaired, but some were still there. We thought, okay, let's wait a little bit longer. 1 week later, we come back. Yes, the number of foci has decreased, but still DNA damage is there. These cells are still not proliferating. And we thought, okay, let's be more patient. And we waited up to 120 days. And as you can see for this cell here, it has still 1, 2, 3 lesions that after 120 days of daily attempts by these cells to repair DNA damage, well, those 3 lesions were not repaired, were not resolved, and that cell is not proliferating because of those 3 lesions that still hold its proliferation. So the simplest interpretation was that, in fact, not all DNA damage is repairable. And we had to study those lesions that persist because they are the only one responsible for the rest and the senescence of those cells, not the other 99% of the lesions, because those are inconsequential. The other 99 can be repaired. So for the cells, having them or not having them is not a problem because they will be able to fix them. Those 3 are special. They are those that dictate the fate of those cells. So we really wanted to understand what was special about those 3 lesions in that cell. That was done in vitro, on a petri dish, on plastic. And of course, I just mentioned before, oxygen. And so you could not really make sure that this was not due to too much plastic, too much oxygen, too much serum, not real, not a real event, not something that happens in vivo. So we extended this observation to a real situation. So this is one of the experiments that we carried out. This is in the mouse brain. We exposed them to ionizing radiation. This is the hippocampus. You can see plenty of these DNA damage response foci. In this case, they are labeled by 53BP1, one of those proteins that I showed you before assembles at sites of DNA damage. And you can see there are plenty of them, but if you let those mice rest and then analyze them 3 months later, after the initial insult, you see that many lesions have been repaired, but not all of them, and still you see a bunch of those. So that means that also in vivo, a small fraction, but an existing, persistent fraction of these lesions cannot be repaired. So this and a number of other results that I'm not gonna show you today, really pointed us to the presence of some irreparable lesions that persist, and therefore they trigger a prolonged activation of the DNA damage response. So what's special about those? Why they are not being repaired? So we consider several hypotheses. One of the wildest was, could it be, there is nothing special about the lesions, it's something special about where they are. Maybe there are some portions of our chromosomes that if broken, they cannot be repaired. So there are sort of black holes in the genome that if damage, which is initially random because X-ray is going to be random, if damage lands there, that region is a no-go for the DNA repair machinery. And we thought, fascinating hypothesis, but probably rubbish, because evolutionarily speaking, it doesn't make you any fitter to have a portion of your chromosomes that if broken will not be fixed. So we were almost about to dismiss this until we realized that, of course, this region would exist and evolution would promote them if there was something good in resisting endogenous DNA repair activities. It would be good if cells did not want to repair those regions, because if those regions were repaired, that would be a problem for the cell. So we started thinking, which could be a region of the chromosomes which is good that it doesn't get repaired, because if it does get repaired, that is a problem for the cell. After some thinking, we realized that we have been working on those regions for quite a while. And these are the telomeres. So I remind you what telomeres are. Telomeres are made of repetitive sequences, all with the same orientation, binding to the same problem binding to the same proteins, all with one main function. And the main function of the telomeres is to prevent chromosomal fusions, because if you have chromosomal fusions, you're going to get tangles at the next mitotic round, and so that is a mess. So telomeres are assemblies of DNA and proteins that prevent chromosomal fusions, okay? But chromosomal fusions are in fact synonyms for DNA repair events. So now you start to appreciate telomeres as inhibitors of DNA repair. For a good reason, they want to avoid this end to be ligated, to be repaired to this other chromosome end, which is fine. The problem in this system is that they are made of repetitive sequences, and as they all cumulatively act at the very tip here, you may start thinking they may be acting throughout their length. So if damage strikes here, this first half of the telomere may start thinking, okay, I'm doing my job today. Today I'm preventing a fusion between this telomere and this telomere. In fact, it is preventing DNA repair within its own chromosome, okay, within its own telomere. So we started thinking maybe those persistent DNA breaks were breaks that were at the telomeres, and since they were unrepaired, they were triggering this persistent DDR activation. So we in fact went back to those experiments that I showed before with those persistent DDR foci, DNA damage response foci, and we localized at the telomeres those foci by co-localizing those markers like gamma H2X, for instance, with a probe that recognizes telomeric sequences. And so we monitored, we quantified these events, and we observed that with time, 4 days, 10 days, 30 days after radiation, there is a progressive decline in the number of foci, because of course cells are repairing DNA damage. This is what you would expect. However, those that persist were progressively found co-localizing with telomeric sequences, okay? So indeed, the longer you wait, the less foci you see, but those few foci that persist are progressively more and more frequently found at the telomeres. So telomeres are getting repaired much less. And this is something specific for the telomeres. It's not something that applies only to, that applies to any repetitive DNA sequence, because for instance, the centromeric locus has a very different behavior, okay? So if I had to, again, simplify in a graphical manner everything that I told you so far, we think that every time DNA is damaged, and there may be exogenous sources of DNA damage, we use X-rays, but damage is constantly being generated by the cells because there are reactive oxygen species, there are DNA replication events, there are transcriptional events, there are topoisomerases that constantly break and do funny stuff to DNA. Well, DNA do break. This always triggers a very active, efficient DNA damage response at all lesions, but this DNA damage response will not be able to repair effectively the lesions that occur at the telomeres. So you end up with repair in the non-telomeric regions and the persistent DDR activation at those unrepaired telomeric breaks. That means that with time, and I'm prone to believe during aging, telomeres fill up with breaks, because as I mentioned before, DNA is constantly being damaged and it is constantly being repaired. But if those lesions happen within telomeres, they will be much less repaired, and therefore they will accumulate progressively these persistent DNA lesions. So in summary, 3 simple messages. Genomes, in fact, are not uniformly repairable. I'm saying genomes because we extended these observations in yeast, in mouse cells, and in human cells. What I like about this story is that this irreparability is really an evolutionary selected trait, which is unavoidably associated with a maintenance of the linear structure of the chromosome. You want to avoid fusions, you're going to accumulate DNA damage. The solution would be to have a circular chromosomes, so no DNA ends to inhibit, but evolution didn't seem to go that way, at least for mammalian chromosomes. And the inference again is that with time, telomeres will accumulate DNA damage, filling up with time, and this may be associated with aging. Personally, I think it is going to be the cause of aging, but of course, determining causality for this event is going to be experimentally hard to demonstrate. So, is then really true that DNA damage fills up at the telomeres during aging? So we had the luck to access to tissue sections of baboons, so primates like us, and we look at tissues which are non-proliferating, like neurons, which are terminally differentiated, and liver, that if baboons do not drink and smoke will be mostly quiescent and non-proliferating. And so therefore there is no replicative senescence, meaning by driven by telomeric shortening. So in those cells that do not proliferate, in which telomeres do not short, do we still accumulate DNA damage during aging? And the answer is yes. If you compare young and old animals, you observe an accumulation of these DNA damage response markers at the telomeres. So indeed, this seems to be going on also in vivo in large mammals, large primates. So in the beginning, I suggested a very appealing way to explain aging proliferating cells. You proliferate, telomeres become too short, DDR is activated, you pull the brakes, which is good to explain aging in proliferating cells, but most of our body is made of non-proliferating cells. They do age, nevertheless. So we provided then a second mechanism, this cell senescence, which is triggered by this random DNA damage, again, also in non-proliferating cells. But we propose that in both cases, there is a shared mechanism, and this is the generation of DNA damage within telomeric repeats that, being reparable, causes a persistent DDR activation. And we are prone to believe that any genotoxic event that will trigger cell senescence will in fact impact on telomeric DNA damage, because by our perspective, if DNA damage is going to be elsewhere, it will be repairable, and if it is going to be repairable, it's going to be only transiently activating the DNA damage response pathways. Okay, so that was about cell senescence in mostly fibroblasts, because this has been the workhorse for many studies in many labs, including ours. But then at some stage we thought, okay, let's study something which is more interesting than boring fibroblasts, and maybe stem cells are cool, even more exciting, neural stem cells. So at some stage I had a postdoc that managed to grow neural stem cells as a monolayer, all very homogeneous, and so we were able to grow them under optimal conditions. And then we did our usual experiment. We take the cells, we expose them to X-rays, and we see what happens. And we observed that yes, many cells undergo apoptosis, so they die, so not unsurprisingly, but not all of them. And those that do not die, they reduce their replication rates, they downregulate a number of proliferative markers, suggesting they were undergoing maybe cellular senescence. So since we are a senescence lab, we immediately run the usual tests. One of them that is the one that I mentioned before, this senescence-associated beta-galactosidase activity, which is probably meaningless, but everybody uses it, so we used it too. And indeed, our cells, we run this assay, and they turn blue, because when this assay works, you get blue cells, and then you count the blue cells. And indeed, you have the vast majority of these irradiated neural stem cells that have become blue, and this could have been the end of the story. So we just discovered that also neural stem cells upon DNA damage undergo senescence, and we were almost about to write up a paper until, and I should really give credit to my postdoc, he told me, Fabrizio, these are the cells before irradiation, these are the cells after irradiation, they look very different. To me, these neural stem cells have become astrocytes. And I told him, I've never seen an astrocyte, you've never seen an astrocyte, we are not a neurobiology lab. So let's run a few tests, let's check for some markers that would really point us toward falsify or verify the hypothesis that these cells upon DNA damage had undergone differentiation. And indeed, he observed that nesting, which is a neural stem cell marker upon DNA damage, was going down, while GFAP, which is a marker of astrocytic differentiation of astrocytes that was absent in these neural stem cells without DNA damage, upon DNA damage was strongly induced. So this made us more confident to put some more effort in this. So we performed a number of analyses. So we analyzed by microarray the expression of a bunch of genes, and we observed that many of them go up, many of them go down, and our collaborators, by analyzing these results, really made us the point that, yes, Fabrizio, you may be right, these cells are undergoing differentiation toward astrocytes, so they are becoming astrocytes. So we were quite intrigued, but of course, this was happening in an incubator on cells growing on plastic. So we were always thinking, maybe this is the mother of all artifacts. So let's check this in vivo. So we designed, we started to collaborate with a group that has some mouse model to prove or disprove our model. And so this is the cartoon that the postdoc drew in order to exemplify the experimental strategy. So basically there is a way in order to trigger a recombination, so a genetic events only in cells expressing SOX2, which are neural stem cells, so that those cells become YFP positive. So you get genetically those cells green. So whatever happens then to those cells, they will remain green. So you have permanently labeled those cells. Then you take these mice with this green stem cells, and then you expose them to X-ray and see if these green cells now start to express also astrocytic differentiation markers. So we did experiments. So these are tissue sections from the brain of these mice before irradiation. So this is the subventricular zone. So ventricles are these holes that we have in the brain, and the cells surrounding these ventricles are known as the cells of the subventricular zone, and they are green because this is where stem cells reside. So this, you see them, and of course they do not express GFAP, this astrocyte marker, because they are stem cells, and they do not express also S1 beta, which is another differentiation marker. But when mice were exposed to ionizing radiation, you see that now these YFP positive cells are now expressing also GFAP, this astrocyte marker, and S1 beta marker. So indeed, also in vivo, it seems that DNA damage triggers the differentiation of these neural stem cells. So our in vitro results were recapitulating events that in fact occur also in vivo. So this is the slide that I showed you before, but maybe at this stage we may feel the need to improve this cartoon, because what we have just observed that in fact there is an additional outcome of DNA damage generation, which is cellular differentiation, and that was in fact my first version of this cartoon, but then I realized it was wrong, because in fact what we observed that those cells were both undergoing differentiation, but they were also senescent, because remember they were both beta-gal positive but also GFAP positive. So in fact a better cartoon is this one, in which cell differentiation coincides with cell differentiation, with cellular senescence coincides with the cell differentiation. In fact, maybe cellular senescence is a form of cellular differentiation that we have all been missing for many years. Why? Because we all started working with fibroblasts, which are already fairly differentiated cells, and upon DNA damage, they will remain fibroblasts because they cannot become anything else. And so maybe if Leonard Hayflick had started his experiments working with stem cells, now we would not be talking about cellular senescence, but maybe we would be talking about DNA damage induced cell differentiation. So this is one way of saying it. So far I've been speaking about cellular senescence and aging, and cellular senescence and aging are certainly strictly connected, but there is a very strong connection to cancer as well. And this is summarized in this cartoon here. So if you take a normal fibroblast, one of those that I was showing you before, and then you express an oncogene, a really angry, nasty oncogene, the one that are typically associated with cancer.
Fabrizio,
yeah.
excuse me, sorry to interrupt you, but I see that you switched to cancer. As
Yeah.
I'm on the other side of the table, say, how pervasive you think is this in the previous slide? So the conclusion, the wonderful, amazing conclusion is, it's a form of cellular differentiation. But do you know from your lab, and I guess other labs becoming intrigued by your findings, do you have a case where you really see that senescence, another case in addition to the astrocytes, where you see senescence?
So there are some examples. And the best example is probably if you take a mouse, you expose it to X-ray, and its fur from brown becomes gray. And why is that? Because people have observed that it becomes gray because the melanocyte stem cell in the hair bulb, upon DNA damage in a p53 dependent manner, undergoes differentiation. And this leads to the loss of pigmentation, and that's why the fur, the hair, becomes gray. So that is one example. There are other examples in the hematopoietic compartment in which upon DNA damage, there is a loss of stemness and the skewing toward myeloproliferation. And so there are at least, as much as I can think of now, at least 3 examples: ours, the hematopoietic stem cell system, and the bulb, in the stem cell melanocytes in the bulb, in which DNA damage has been shown to be associated with the cell senescence and differentiation. Yeah.
Fabrizio, on a personal note, since my hair is gray since I was 25 years old or so, well, since today I can think that after all there is DNA damage involved in that. So, you know. So
that means, yeah, there is some broken DNA in your bulbs somewhere.
Let's hope for the best.
Yeah, but don't think about it.
No.
So that was really my, I mean, are there any questions from the students on this? We'll come back to aging, but for a while we're gonna do something else. Okay, so if there are no more questions, yeah, or maybe there are.
Can I ask something?
Yeah.
I was wondering why the differentiation in the cell is induced. Like way to the cell to survive. It's a survival tool of the cell, or is just an induction?
No, that's a good point. So why the cells decide to differentiate? Maybe again, it's a defensive mechanism. Well, number one, a stem cell has a lot of potential and can become many things, and can proliferate and can differentiate, but a broken stem cell is not a good idea to have around. So maybe it is safer to be quiet, become differentiated, stay put, and don't do much damage, because you are a cell that you have problems. So you better not proliferate and expand your progeny with your broken DNA. So that may be the simplest interpretation I can give you.
Okay, thank you.
Okay, so
sorry, in this case we lose the pool of stem cells.
Yeah.
Okay.
Yeah, indeed. Yeah, that
Thanks.
is what we are thinking is going on. So we were saying cancer. So cancer and cellular senescence. Well, if you take a normal fibroblast and then you express a very aggressive, very powerful, very potent oncogene like RAS, HRAS-V12, it's very aggressive, expressed in a number of tumors. Well, if you put such an oncogene in a normal cell, you would not get cancer, you would not get transformation, you would not get wild proliferation, you would get, in a matter of days, senescent cells. So clearly this is a defense mechanism, once again, that prevents the expansion of oncogene expressing cells. So it's tumor suppressive mechanism. And these are, again, many of the oncogenes that have been associated with these events. So what happens then, in fact, when you express an oncogene? And this is something that we have studied. in the lab. It was one of the first lines of research when I arrived in IFO in Milan. And so we did a very simple experiment. So we started counting cells upon expression of an oncogene. And so we use a viral vector to express this oncogene. So we use either an empty vector, and you see this exponential growth of cells that grow and grow and grow happily when you don't express anything meaningful. Or what happens when you express in the same cell an oncogene with the same vector. And we observe that initially cells proliferate much faster, which makes sense. These cells are expressing an oncogene, something that should push cells to proliferate very fast. But then cells start to slow down, and eventually they senesce. So yes, they do try to proliferate much faster, and then something goes on. There is a brake being pulled that prevents this proliferation. And of course, what we immediately went to check was to study whether the DNA damage response was activated. Indeed, we observed there was a very neat correlation between the slowing down this hyperproliferation and the activation of DNA damage checkpoint response activation. And we demonstrated causality in this system by removing these brakes, by removing these checkpoints. And we observed that now these cells keep proliferating exponentially because there is nothing that restrains them. And then if you take these cells and impute them in mice to check whether they can generate a tumor or not, what we observed that these cells indeed, when injected in the flanks of naked mice, you see that they make tumors while these cells are unable to generate tumors. So indeed, this really proves that oncogene activation leads to cellular senescence, which is a tumor suppressive mechanism. And then if these tumor suppressive mechanisms are not in place, tumors grow unrestrained and keep proliferating. Indeed, oncogenes break DNA, and this is an example. These cells have not been exogenously irradiated. In these cells, we have just expressed an oncogene. You can see how much damage is being generated. So here we have stained them with a bunch of antibodies against many different DDR markers, but the message is very simple. There is a lot of broken DNA in these cells, again, broken by the expression of an oncogene. And we also demonstrated these oncogenes were breaking DNA by altering the DNA replication process, okay? So they make cells proliferate too fast, and when you run too fast, you stumble, and then you break your leg. Again, we were keen to check that this is not happening in an incubator in IFOM, but it does happen in real tumors, and these are normal and tumoral counterparts of different tissues, different organs, lung, breast, kidney, colon. And as you can see, very often there is an activation of these DNA damage response markers in the tumoral counterparts rather than in the normal counterparts, okay? So indeed, in tumor evolution, there is the generation of DNA damage and the activation of the DNA damage response. And so we were happy, and we were studying cellular senescence, and we were recognized by the field, by the community, and people believed us. And then this happened. So we stumbled in the prototypical elephant in the room. So we recognized that in the room of the DNA damage response field, there was a big elephant that we couldn't see, but we could touch, we could smell, and when we realized there was an elephant in this room, we could not study the elephant. So we had to switch from an established lab studying cellular senescence in studying something very, very weird and unexpected, which took us a lot of time and energy, and to an extent frustration. But it was worth it. So what we discovered that there is something missing in this slide, without which you would not get anything that I've described so far. So we discovered that sites of DNA damage, you need to synthesize and process RNA, which are generated at the sites of DNA damage, because if you don't do that, you don't get the DDR foci formation, you do not get transient checkpoint enforcement, you do not get the establishment of cellular senescence. So for many years, we had to study these weird events. And again, we're not an RNA lab. Certainly, they're not RNA species easy to study. So it was painful. So rather than go through all the grievance and tell you how painful it is, I just simplify things in an animated cartoon, which is the next slide, that again synthesizes a lot of efforts. So what we discovered is that wherever a DNA breaks, this is sensed by the mRNA complex, the Mar11, Rad50, MBS1 complex that I mentioned before. Okay, so far so good. This is what the canonical view would tell you. This is what is happening. But then what we discovered is that mRNA binds and loads at sites of DNA damage RNA pol 2. And this is an active RNA polymerase 2 that will transcribe bidirectionally what we called damage-induced long non-coding RNA. They are more abundant from the break, but we see them induced also going toward the breaks. So double-strand breaks trigger local transcription. Now, these transcripts are further processed by endoribonucleases called the Drosch-Dicer that chops this long RNA into shorter ones that we call the ddRNA. And then we could demonstrate that the small ddRNA bind by sequence complementarity to the complementary precursors. And only when the small RNA are bound to the long complementary RNA, they allow the formation of this DDR foci that I showed you before. 53BB1 is one of the components of the foci that we have been studying for longer. So the simple message is that indeed the double-strand breaks are in fact transcriptional promoters, bidirectional transcriptional promoter, because they promote the transcription starting from the break. Then one question that for the last 2 years we have been asking the lab, so is it really true? So is it really a double-strand break pretty much like a transcriptional promoter? So we had to go back to the textbooks because we are not a transcription lab. And so we had to study how transcriptional promoters are assembled, and RNA pol 2 is not traveling alone. It's not sitting in the genome anywhere if it is not positioned and oriented and activated by a so-called pre-initiation complex, which is a huge complex of protein made of additional subcomplexes, and you may have heard of TF2D, TF2B, TF2E, H. For instance, the Tata box binding protein, TBP, is one of the components of TF2D. And so canonical promoters will have the pre-initiation complex sitting on the DNA and then recruiting, loading, and activating RNA pol 2. So we asked recently whether also the pre-initiation complex was being loaded and recruited to double-strand breaks or not. If not, fine, that means there is a difference between transcriptional promoters and double-strand breaks. So we did a very simple experiment. We introduced a double-strand break in an untranscribed region. So we break this region, and then we monitor by chromatin immunoprecipitations the recruitment there, not only of RNA pol 2, but also of a bunch of components of that pre-initiation complex. And we monitor their association at different distances from the break, okay? And we observe, so it's very close to the break, 100 bases on the right and on the left. I'm just gonna show you on the right. We observed that as soon as you cut, the gray bars are upon induction of the cut, okay? You get gamma H2X phosphorylation, as you expect. You get a recruitment of RNA pol 2, not of RNA pol 1, not RNA pol 3, fine. But you also recruit TBP, TF2A, B, E, FH, mediator 1, CDK7, CDK9. I will not go through the details, but pretty much everything that people usually find at transcriptional promoters, we also find it to be associated with double-strand breaks, okay? So it really looks like a transcriptional promoter, but this is very close to the break. Further away, you just see RNA pol 2, not the pre-initiation complex. Even further than that, you just get gamma H2X, not even transcription, not even RNA pol 2. So this really looks like a gene unit. You have a promoter, you have a transcribe unit, a transcribe region, and downstream 3' from it, not even that. So it really looks like a gene. And so now we are working in this direction. We're checking whether this synthesis of non-coding RNA can actually generate proteins if this double-strand breaks is occurring in a region upstream of a doping reading frame. But this is something for maybe the next seminar. So yeah, the answer is yes. So really, double-strand breaks really are very similar to transcriptional promoters. And then the next question is, why the hell cells put so much effort in synthesizing RNAs at sites of DNA damage which are non-coding? Because of course they transcribe the locus which is broken in that moment. And this RNA will always be different because wherever DNA breaks, that region will be transcribed. So each RNA will be different. Well, what we discovered recently is that this D-link RNA and ddRNA allow the formation of these foci by trapping and condensing the proteins that are otherwise would be broadly and diffusely distributed to the throughout the nucleus, and they assemble rather in these blobs, these foci, and here this assembly of proteins and RNA, which are differently called, people call them liquid phase separation events, or liquid demixing, or condensates, or yesterday I even heard another word that I've already forgotten. And because this is something really new, and this the field is still evolving, hasn't settled on a term. But the message here is that the RNA is there in order to retain those proteins around the sites of DNA damage to form those foci. Indeed, these foci have a globular structure, so they have a shape, but they don't have a membrane to keep the proteins around the break. So they are membraneless globular structures, and they are totally dependent on RNA, because we demonstrated that if you prevent RNA-APOL2 activity, you prevent their formation. But actually, even when they are they are already formed, if you add some RNAs that degrades the RNA, the foci melt and they disappear. So we characterize a lot the features of these foci, and this is really biophysics. So you started a collaboration with some physicists in Milan, and for us, they told us that the way that these foci evolve in time is really consistent with them being sort of liquid. So initially you have these foci that then they grow in size and then start to coarsening, meaning that they start to merge and then fuse together. And probably the easiest way to convince you that these are not static, rigid, stiff, still structures, but very dynamic, mobile, liquid, is to show you how wobbly these are. So I'm going to show you a movie of 40 seconds of the life of a 53-big-1 focus, and you can see how dynamic it is. It's really, really wobbly. And of course, our physicists were very good in calculating the ripples and the capillary waves on these structures. And so they were very happy to draw equations that I never really fully understood. But eventually we could really demonstrate that these structures are indeed liquid. They are viscous, but liquid, and they have the same liquidity of honey, which is the only concept that I managed to grab out of their equations. So they are liquid structures, and they are held together by RNA. That is the message for you guys. So let me go back to this point here, because I think this is crucial for the rest of the presentation today. What I told you is that you break DNA and you get these RNA that are necessary to hold proteins together. If you look at this staining, you see the same proteins everywhere: 53-big-1, 53-big-1, 50. So all the foci look the same. However, if the broken DNA leads to the synthesis of this non-coding RNA, each of these foci contains a different RNA. because this RNA is the result of the transcription of that damaged locus. So the RNA is the only component which is unique, which is different in any individual focus, okay? So if you want to inhibit DR everywhere in the cell, you invent a 53BP1 inhibitor, you're gonna be very effective, but you're gonna be non-specific, meaning you're gonna inhibit it everywhere in the nucleus. Differently, if you manage to inhibit the function of the RNA in this focus here, for instance, because you know the sequence, for instance, then you will be able to inhibit DDR formation at that focus here, while not altering DDR activation elsewhere. So, which is a way to control, inhibit, interfere with the functions of this non-coding RNA. So we thought that we should design antisense oligos which bind to this RNA and somehow inhibit their functions. And indeed, I'm not going to show you the results, just going to show you here in this cartoon what happens. We observed that this antisense oligonucleotide, by binding and competing for the RNA-RNA interaction that I showed before, they dismantle this foci. And we could demonstrate that, and they inhibit DDR in a sequence-specific manner, okay? So, in fact, this really suggests that we can inhibit DDR in a sequence-specific manner, okay? And this now, maybe I should ask you, Andrea. So I think I have 10, 15 minutes, which I'm afraid just bring us straight to 11. So maybe we should just push forward and go till the end of the lecture without breaks. What do you think?
Up to you. I think they probably have to leave at 11.
Yeah, exactly.
So I think if you can manage in 5 minutes.
Yeah. Okay. So I'll wrap it up. So if there is time for additional questions. So can we inhibit DDR in a sequence-specific manner? And anyway, what for? Well, I go back to the beginning of my lecture today. So telomeres accumulate DNA damage during aging. And so how about inhibiting DDR activation at the telomeres, and therefore inhibiting cellular senescence and therefore inhibiting aging? And so the idea is that these are the telomeres and they are damaged for whatever reason, and this may lead to the synthesis of this non-coding RNA. We call them telu G and telu C because they are on the G strand or the C strand of the telomeres. And then we're going to use antisense oligonucleotide to inhibit them by sequence complementarity and kill their functions. And we tested them in many different ways. I'm going to show you 2 examples. The first example, well, more than the first example is a mouse model in which I remove one of the proteins that sits on the telomeres. It's called the TRF2. Without TRF2, telomeres are exposed DNA ends and they trigger very strong DNA damage response. So I do remove TRF2, but at the same time, I inject mice with this antisense oligonucleotide against this telomeric non-coding RNA. And what we have observed that in these mice, we have a very strong DNA damage response activation, but downstream of gamma H2X, when I inhibit DDR with this antisense oligonucleotide, again, the anti-telu G or anti-telu C, you can see here that DDR is strongly inhibited, and this is the quantification. So yes, we can inhibit DDR in a sequence-specific manner. The DDR telomeres can be inhibited with these antisense oligos. And so we think we want to study aging. We want to study the contribution of telomeric DNA damage and telomeric DDR activation into aging. Aging is interesting, but accelerated aging is even more interesting. There are a number of conditions that cause this accelerated aging. Hutchinson-Gilford progeria syndrome is one of them. It's caused by many different, it is associated with many problems, among which telomere dysfunction. And Sami Basso is the longest living HGPS patient. He was 22 when we took the picture, now he's 25. And of course, it doesn't look 22. It looks like a very old person. So probably for the sake of time, I'll skip you the in vitro data. I'll just go to the in vivo data. So we treated mice carrying the same mutation of Sami Basso with these antisense oligonucleotides. These mice have this mutation only in their skin. So we are looking only at the skin of these mice. And these mice treated with our antisense oligonucleotide, we look at DNA damage response activation. These are normal skin. These are the skin of mice treated mice with a HGPS mutation. You see the DNA damage, response activation, 53bp1, phospho-Cap1 marker. That in these mice treated with this antisense oligonucleotide to a good extent, you see a reduction of this DDR activation. We see a reduction of P16, which is also a marker of cellular senescence, a driver of cellular senescence. We see less apoptosis, we see less infiltration of immune cells, because again, we see less induction of an interleukin that we know is associated with cellular senescence, that we know we can reduce by killing the DNA damage response with this antisense oligonucleotide. But what is important is that in this mouse model, and these mice never ever lived longer than 9 days, with a treatment with this antisense oligonucleotide, that they lived 50% maximum survival more. Do they live like wild type mice? Surely not. Do we make a statistically significant, but also biologically relevant impact on their biology, on their lifespan? We think so. And to the least, we think that this proves that telomeric DNA damage makes a contribution in this disease, and of course that these may be more than tools for researchers, and these antisense oligos may have an impact also in human conditions. So now we are extending, since this was, as you can see here, this was occurring, everything was occurring in 10 days, so you may say, very short, so aging is lower and more impactful set of events. So we used a different mouse model in which telomeres are shortened because we remove telomerase, we treat mice when they're young, and then we analyze them 10 months later. So this is a much longer term impact that we are studying here. Again, I will not go through all the details. These are just look at the lungs. These are lungs of wild type mice. These are lungs without telomerase. You see how compromised. There are very few holes in these surfaces. This is where oxygen and CO2 are exchanged. Less air spaces, less breathing for these mice. But upon treatment, you see that to a good extent, these air spaces are improved. And this is strongly related to our ability to reduce DDR activation, as you can quantify here. And so we are extending this observation in these mice. We are also studying zebrafish, which is another excellent model for aging. We are using telomerase mutant zebrafishes, and we see that we can improve their fertility, we reduce the developmental abnormalities, and also here we are impacting in an important way on their survival. So again, maybe I'm being very fast for the last part, but again, this is the last slide that summarizes everything. We think that with this approach, based on antisense oligos against this telomeric non-coding RNA, we can inhibit the DDR at this damaged tumors, and we can restore a tissue homeostasis and extend the lifespan in vivo in different animal models, and importantly in different species. I showed you mice, I showed you zebrafish, and what I really want to stress, even if they are triggered by distinct progenitor genetic defects, meaning that this is not a treatment for a specific mutation in a specific gene, anything that will eventually impact on the telomere biology, anything that will eventually lead to telomeric DDR activation. If telomeric DDR activation is one of the causes of the condition, of the disorder, of the disease, this approach may turn out to be beneficial. So I'll stop here. This is us, but in fact I was not sure how, because I showed you really many years of work. So and the lab has evolved with time. So this was us a few years back, and then we took additional pictures. So this was in an escape room from which we didn't escape. And this is the most recent one is this one. And even that is not updated because in fact a former student from Andrea is not in. So I have to generate an additional one. So I'll stop here with some relaxing images of 53bp1 foci for you to relax and ask me questions. So I'll stop here.
Grazie Fabrizio. Grazie tantissimo. I think there is time for a couple of questions or domande. È stata una bellissima finale per il corso di biologia molecolare della triennale, perché tutti gli argomenti, buona parte degli argomenti che ho fatto a lezione sono stati toccati da questo seminario, quindi per me è stato
Bene.
Ciao. Forse avete almeno una domanda da fare. One question. Not people may...
I have a question. May I ask?
Sure.
Okay, now when we induce senescence with drugs, so in this case, we don't have, let's say, damage in telomeres. There is we can study this kind of senescence, comparing it with the senescence derived from the damage response, or it could be different as a study model.
I'm not sure I got the question. Say it again, sorry.
No, because I was wondering about drugs that induce senescence.
Mm-hmm.
I don't know, maybe palbociclib can be an example.
Mm-hmm.
So acting at the cellular level in the division of the cells. So in this case, we don't have probably damage in the telomeres. So can we use this model as the model of senescence induced by DNA damage, or it could be different?
Yeah, no, everything really boils down to the fact whether DNA damage would be generated or not. What I can say is that when we use different chemotherapeutic agents like bleomycin, for instance, that is also generating DNA damage, but not specifically at the telomeres, we did observe the accumulation of DNA damage at the telomeres, and therefore the model that I'm proposing is also based on the evidence of DNA damage generated by bleomycin. We didn't try all the different kind of reagents or treatments. Maybe it's fair to say that our model is probably relevant in case of genotoxic induced cellular senescence. If cellular senescence is induced by some other treatment that seemingly does not trigger any DNA damage, likely our model is probably useless and additional mechanisms are in place.
Yes,
but again, I am a DNA damage centric and I'm telomeric DNA damage centric. So I always think that eventually DNA damage is at the root of everything, but occasionally I'm wrong. So.
No, just to understand, because maybe we induce senescence at a later point, so maybe acting on the cell cycle directly, and so we lose the DNA damage.
Yeah. If you're acting if you act downstream, you act downstream. So basically, you mimic the consequences of a persistent DNA damage. Yes.
Okay. Thanks a lot.
That is correct.
So I think it's probably time to end this wonderful event.
I have a quick question, if it's okay.
Yes. Yeah,
sure.
Thank you so much for your presentation. It was extremely interesting. And you mentioned this progeroid syndrome, like early aging. Do you think that your research will be beneficial for the patients?
This is what we are thinking. So it is honest intellectually to admit that this treatment does not fix the problem, fix some of the consequences of the problem. So we think that maybe for HGPS, this is probably, I would not prioritize it among the treatments. So people are now using antisense oligos in order to bypass the mutation. It's called exon skipping. And this may have more better, maybe a better option for the patients, but there are many other conditions. I showed you the lungs of those mice. So there are a number of conditions associated with the telomeric shortening and telomeric DNA damage. For instance, pulmonary fibrosis is one of them. It happens in people when they are old and make them unable to breathe. And there is a very well-established link between telomeric DNA damage, DDR activation, fibrosis, and this disease. So that, for instance, is an option. It's a disease that we think we could make a difference with this treatment, and this is the direction in which we are going now. Yeah.
Fantastic, thank you.
Excuse me, may I ask you a quick question?
Yep.
You talked about the role of double-induced long non-coding RNAs. Has this role been discovered quite recently?
Yes, very. So 5 years ago, we were the first, yeah.
Okay, thank you so much.
May I add a quick curiosity? It's not a question, but it's a technical issue maybe.
Yeah.
So thinking about the future possible therapy, how these oligos are delivered to the specific tissue we want to target in a, thinking about a real therapy in a real patient.
Okay, so some oligos with the same chemistry are already FDA approved. So these are not just, it's not only a weird approach, this is something that is already happening for other diseases with other oligos, with other sequences, but the chemistry is the same. You may, in the case of lung, you may inhale oligos, and there are already devices and approaches and clinical trials in this direction. You may also conjugate with some antibodies or some other moieties that deliver the oligo specifically to a certain organ, to a certain tissue, to a certain cell type. So there is a way to, to improve the delivery, the specificity of the delivery of these antisense oligos, or you may also consider a broad distribution, because when you take an aspirin, you get a relief of a headache, but the aspirin will go throughout your body. And so you may also consider a systemic delivery.
Thank you.
No more questions. So for the MBG students, see you tomorrow. The molecular, see you at the exam. And once again, Fabrizio, thank you so much. Elisa, thank you so much to you as well. Elisa, vuoi dire qualcosa?
Ma il tempo è tiranno, quindi direi che li lasciamo andare. Vi ringrazio te, ringrazio Fabrizio. Magari ti lascio in separata sede la presentazione, i contatti, il questionario, quindi poi se hai modo a posteriore di condividerli con i ragazzi, adesso li lasciamo andare, insomma.
Ti possono dare anche gli indirizzi mail direttamente.
Ah, fantastico, ok.
Grazie a tutti, see you soon.
Grazie a tutti, grazie, ciao.
Ciao ciao.
Grazie.
Thank you, bye.
Thank you.